everybody, my name is Becca Klopp and I'm a graduate student at Clemson University, and today I will be talking about brown fat in newborn animals and thermoregulation. Just to kind of start off, I'm going to do a little bit of a review of what we talked about in class already, just so that we're kind of all on the same page for the rest of the presentation. So there are three different types of adipose tissue. There is brown adipose tissue, which is also referred to as fat or brown fat. There is white adipose tissue, which is also referred to as WAT or white fat. And then there's beige adipose tissue, which is also referred to as bright, which stands for brown in white. From the picture that I have on this slide, you can see that the white adipocyte is mainly made up of a lipid droplet. The brown adipocyte has lipid droplets throughout, but it also has mitochondria. And then the bright adipocyte is kind of a mixture of the two of them. So we talked a little bit about the uncoupling protein 1 or UCP1 in class, and this is an intermitochondrial membrane protein. When it's activated, it allows the free flow of protons across the intermitochondrial membrane, and this process causes the rapid dissipation of chemical energy as heat. And then this heat that is produced from this method is referred to as non-shivering thermogenesis, which basically is exactly like it sounds. A lot of times when you're cold and you're trying to get your body temperature up, you shiver, and in this process, it's another way that you don't have to shiver. It maintains your body temperature without having to shiver to stay warm. So it is very important in maintaining body temperature in newborn animals. This ability is usually lost in animals like sheep. It's usually lost about 7 to 10 days after birth. In other animals, it's replaced with white adipose tissue or beige adipose tissue. And then in humans, we do still have some brown adipose tissue that is present, but it is not as active. And then in pigs, pigs do not have brown adipose tissue. Next, I'm going to talk about the mechanism of brown adipose tissue and the uncoupling protein 1. So brown adipose tissue does contain a very large amount of the uncoupling protein 1. Just to kind of put it into perspective, when UCP1 is kind of at its max or its peaks, it can produce up to 300 watts per kilogram, and this compared to other tissues that might be able to produce about 1 watt per kilogram. So it can produce about 300 times as much heat as other tissues, so it's a lot more. This process is regulated by the unmasking of GDP binding sites that are located in the uncoupling protein 1. All processes like this within the body need some kind of energy source, and this process gets its energy from non-esterified fatty acids or free fatty acids from lipids, and this process is activated by the sympathetic nervous system. We'll talk a little bit more about that later on in the lecture. I put this picture on here just to kind of show the very, very basic of what is going on here and what's happening to the protons. So as you can see on the inner membrane space side, there's two different processes, and they both start with protons. And normally protons will go through ATP synthase and be added to ADP to become ATP. But if UCP1 is activated, these protons that are in the inner membrane space because of the electron transport chain, they instead will go through the UCP1 and produce heat. So just kind of a basic overview. Again, it's activated by the sympathetic nervous system. The energy source comes from free fatty acids. The unmasking of the GDP binding sites is what regulates this. This causes the free flow of protons across the mitochondrial membrane via the UCP1, and this produces heat. Brown adipose tissue does differ depending on the species. Non-shivering thermogenesis does occur in a majority of mammals, including humans. A lot of times you'll see this present in precocial animals, and precocial animals are kind of born and they're more mature, more mobile, They're not as helpless as nutritional animals, and they do not benefit from huddling with their litter mates, so they rely heavily on non-shivering thermogenesis. Humans do contain a large amount of brown adipose tissue, and brown adipose tissue is kind of located around the internal organs and major veins and arteries, which if you think about it makes sense, because if your body temperature goes down, a lot of times your organs can start to fail and blood flow will decrease. So it makes sense that this is located in those areas to make sure that the body temperature stays up and your organs are fully functional. Mice and rats have a very short gestation, so you're looking at about 20 to 24 days, so it's not very long at all. So when mice and rats are born, they have an immature HPA axis, which is the hypothalamic pituitary adrenal axis, and this is responsible for a lot of endocrine signals. And as you can see, I just kind of put a picture up here of how one hormone released by the hypothalamus affects the anterior pituitary, which a hormone that releases affects the adrenal cortex, and then there's negative feedback in all of them, so they're all interrelated. And this axis is not mature in mice. And because it's not mature, then therefore brown adipose tissue is not mature, and it doesn't mature until later after they are born. So mice and rats have to huddle with the other pups in order to stay warm. And the sheep, which have a longer gestation, about 142 to 152 days, do have a mature HPA axis prior to birth. So when they are born, brown adipose tissue is fully functional and UCP1 is active. There is research that has shown that in sheep, the HPA axis does kind of mature late in gestation. So if sheep are born preterm, sometimes they won't have a mature HPA axis, they won't have mature brown adipose tissue or UCP1, and therefore they can suffer from hypothermia. And then pigs, like I mentioned before, they do lack brown adipose tissue, and this is because they have a non-functioning UCP1 gene, and this was caused by several different mutations, so pigs have to rely completely on shivering thermoregulation in order to maintain their body temperatures. Next, I'm going to talk a little bit about the development of brown adipose tissue, and for this example, I'm going to be talking specifically about sheep, because sheep are kind of the most studied animal model when it comes to brown adipose tissue. So this example that I have is referring specifically to them. But in sheep, about mid-gestation, which is approximately 80 days, there is brown adipocytes present. They're visible, but they do not express UCP1 yet. And then at late gestation, about 140 days, we see that compared to mid-gestation, adipocytes are larger in size, and they now contain functional UCP1. And this time point is kind of when UCP1 activity peaks. And then at birth, we already start to see a slight reduction in the number of adipocytes and the expression of UCP1. About a week old, we see an even further reduction in the number of adipocytes and the expression of UCP1, and then at about a month old, brown adipose tissue in sheep is completely gone. White adipose tissue remains, and there is no UCP1 present. I'm going to go a little bit more in depth about the activation of UCP1, and like I mentioned before, it is activated by the sympathetic nervous system, but there are some specific stimuli that activate the uncoupling protein 1 and non-shivering thermogenesis at birth. So this picture that I got from a study that looked at sheep, and we're just going to start at the top with birth itself. So this process of the animal being squeezed through the birth canal is going to increase prolactin and cortisol, and this is going to help to stimulate the HPA axis, which in return will stimulate uncoupling protein 1 and non-shivering thermogenesis. Also, increased muscular activity has a role in activating non-shivering thermogenesis, and it increases the hormone irisin. Cold exposure in itself will increase increase the thyroid hormones, which will stimulate the sympathetic nervous system and increase non-shivering thermogenesis. Cardiac function will also do this at birth. All of these different things are going to help to increase the activation of this regulation. And then also milk intake is going to increase prolactin and cortisol, and this is going to increase the liver function or hepatic function. So all of these are kind of working together to make sure that this animal is experiencing non-shivering thermogenesis and its body temperature is regulated. And all of these hormone concentrations do decrease in sheep after about the first week of age. So you'll see non-shivering thermogenesis decrease, and then shivering will be the dominant cold response. There's a lot of research out there about brown adipose tissue. Research looking at it in different species, different processes in the body that can affect it. There's a lot out there. So there's no way that I can even scrape the surface on everything. So I wanted to just cover the basics in this lecture, and I found this research paper and I thought that it would be really good to talk about just because it kind of shows that even within species there can be differences when it comes to brown adipose tissue. So this paper, it's not new. It was done in 1997. It's from the Journal of Animal Science, and this project was conducted at Texas A&M, and it's titled Genotypic Effects on Norepinephrine-Induced Changes in Thermogenesis, Metabolic Hormones, and Metabolites in Newborn Calves. And basically, they measured the heat production in newborn Angus, Brahman, and totally sired calves born to Angus and Brahman dams before and after norepinephrine infusion. So there's 3 different dam breeds, or 3 different sire breeds and 2 different dam breeds, which I kind of outlined at the bottom. So you're going to have Angus, Angus, Brahman, Angus, totally Angus, Angus, Brahman, 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 and totally Brahman calves. And basically this project just wanted to see if there was a genetic effect on brown adipose tissue and thermogenesis. And what they found was that calves born to Brahman dams, so sire didn't have as much of an effect, but the calves that were born to Brahman dams compared to Angus dams had brown adipose tissue that was thermogenically less active. And they thought that this could be why calves with Bos indicus inheritance have less cold tolerance. So just in case you don't know, with cattle there are kind of 2 subspecies. There's Bos indicus and Bos taurus. Your Bos taurus are going to be your Angus, your Hereford, Seminoles, and your dairy breeds, and they originated in Western Asia and are usually referred to as European or continental breeds. And then Bos indicus is going to be Brahman, and they originated from South Asia. So even within kind of species, you can see that there is some genetic effect on brown adipose tissue. And like I said, this study found that calves that have Bos indicus inheritance might not be as tolerant to the cold as calves that have Bos taurus inheritance. I'm going to end this presentation talking about the future research topics of brown adipose tissue. So a big one that's kind of going around right now is brown adipose tissue and its role in energy balance in adults. So the idea, like if you think back to the diagram that I showed you with the sheep and all the different kind of pathways that help activate the sympathetic nervous system and brown adipose tissue and the uncoupling protein 1, there are many different pathways kind of that help to promote this and the non-shivering thermoregulation. So the idea is that, especially in humans who we still have brown adipose tissue, it's just not near as active, if there's a way that we can kind of reverse brown adipose tissue to become more active, or for in species like sheep where it completely disappears and is replaced with white adipose tissue, if we can kind of reverse it back to brown adipose tissue. And the idea is that if we do this, we can really help decrease obesity, especially in humans. And most of the research in this area has been done with just mice and rats, so nothing really in larger animals or humans. But like I said, the idea is that this could help decrease obesity because as you as you've seen through this presentation, that brown adipose tissue and uncoupling protein 1 has the ability to release large amounts of heat. And this could just be a process that if humans have very active brown adipose tissue and UCP1, that they could help reduce the excess energy in their bodies and release it as heat. And then I just put a diagram over here of kind of all the different other health effects that come from obesity. So by decreasing obesity, we would be kind of decreasing all of these other diseases, whether it be heart disease, high blood pressure, cancer, liver disease, anything like that. Another area of future research for brown adipose tissue is just looking at this further in different species. Because like I said, there is a lot of variation between species, so just kind of knowing all the differences between all the different animals out there. And then last, just if there's any other factors that help to stimulate brown adipose tissue. Again, like that diagram with the sheep of all the different pathways. If there's more pathways and more stimuli at birth or later in life or diet that help to increase brown adipose tissue, or if there's external factors, we talked about temperature and maybe daylight or anything like that, how internal and external factors can affect brown adipose tissue. These are the references that I used for this presentation. If anybody has any questions, feel free to contact me on Canvas or to leave a comment down below on YouTube, and I will be very glad to answer any questions that I can. Thank you everybody for listening.